perfectly so that each new daughter cell that results from binary fission is going to have a complete chromosome, right?
Yeah.
So the whole purpose of having that DNA for a bacteria just like us is to have blueprints, right? A set of instructions on how you build every single protein. So that's what we're going to do today. We're going to talk about how bacteria take that DNA and they use it as a set of instructions to build a protein. So we're going to go through it piece by piece. So this is going to be a structure of a gene, right? So we're going to talk about this in a second. Guarantee question on exam number 3. I'm going to give you a sequence of DNA very similar to this, and I'm going to ask you what protein is going to be made from this. So you got to transcribe it, then you got to translate it, and so we're going to do that together. Okay? That's where we're headed. So we talked about this a little bit early on, right? The very general steps of protein expression, right? So we have our gene, which is a very short segment of DNA. We're going to transcribe that. Anybody remember what enzyme is responsible for transcription?
Reverse transcriptase.
So it does do that, but it goes the other way for HIV. What about plain old normal transcription?
mRNA.
It makes mRNA.
DNA polymerase?
RNA polymerase.
It's a polymerase, but it's making RNA. So RNA polymerase. So RNA polymerase, its job is transcription.
We're like just naming off everything.
Well, you were all like super close, which is awesome, right? So RNA polymerase transcribes a gene into RNA. This says messenger RNA. That's totally true, but we're going to complicate that today. RNA polymerase actually makes 3 different kinds of RNA, one of which is messenger RNA. Messenger RNA specifically is what is brought out into the ribosome if you're a eukaryotic cell, but if you're a bacteria, it's all one big open space, right? Because there's no membrane-bound organelles. So the ribosomes will take that messenger RNA and translate it into protein. So that's what we want to do. How do we go from here to here? So the first step is transcription. The second step is translation. So we'll talk about transcription first, how that actually happens. So this is a big old complicated picture, right? So we're not going to worry about that picture here. We're going to come back to it. What I'm going to do is show you an animation. These animations are awesome, right? If you went to college back in the dark ages like I did in the late 1990s, early 2000s. So I'm going to show you this animation here, and this is going to be transcription, and then we're going to transcribe a gene.
Let's look at a single gene, which consists of a stretch of DNA with some specific control sequences. Near the start of the gene is a region called a promoter. Near the end of the gene is a region called the terminator. The sequence between the promoter and the terminator is the part of the gene that codes for the protein being produced. Transcription begins.
Okay, so we're going to stop right there. So this is nothing but a long sequence of DNA. This is the bacteria's chromosome, right? So this is, in most cases, just a big circular DNA molecule that for E. coli is about 4.2 million base pairs long. This could be like 2,000 base pairs of it. So on one E. coli genome of 4.2 million base pairs, remember last time, there's roughly around 4,300 genes. So genes are just much shorter sections of DNA, but each one of them has the instructions on how you build one protein. But this is a term we really haven't used before. So what the heck is a promoter, right? So think about this. RNA polymerase we just talked about is the enzyme that's responsible for transcribing a gene. It's the one that undergoes the process of transcription. So when RNA polymerase comes up to the DNA, how does it know where to start transcription? And the answer to that is it finds the promoter. The promoter is a sequence of DNA nucleotides that tells RNA polymerase where to start transcribing. So then RNA polymerase is going to transcribe all the way through the gene. If it doesn't stop, it's going to go in a giant circle of 4.2 million base pairs and get right back to where it was, which would be a real waste of energy and time. It wants to stop at the end of the gene. So how does RNA polymerase know where the end of the gene is? How does RNA polymerase know where to stop? And the answer to that is on the other side of the gene is a termination site. All it is a sequence of DNA nucleotides, but it has just the right sequence that tells RNA polymerase you're at the end, stop. So we're going to have a messenger RNA that starts at the end of the promoter and ends at the beginning of the terminator. That's the gene. That's the part that's actually going to be transcribed into messenger RNA. The promoter tells RNA polymerase where to start. The terminator tells it where to stop.
Is that the start codon, or is it before the start codon?
Start codon is the 3 nucleotides that are blue.
Gotcha.
That's a good question. It's like the promoter is a sequence of DNA nucleotides that tells RNA polymerase where to start, but that's not included in the messenger RNA. It's land here and then start. Okay. Let's start this thing all the way over.
Let's look at a single gene, which consists of a stretch of DNA with some specific control sequences. Near the start of the gene is a region called a promoter. Near the end of the gene is a region called the terminator. The sequence between the promoter and the terminator is the part of the gene that codes for the protein being produced. Transcription begins with initiation, when a molecule of RNA polymerase binds to the promoter. The promoter contains a specific sequence of DNA bases that is recognized by the RNA polymerase, allowing it to bind. Notice that the sequence of the promoter plays an important role, even though it isn't part of the protein coding region of the gene. The RNA polymerase makes its way along the gene, synthesizing the RNA molecule from the template strand of the DNA. Elongation continues until RNA polymerase reaches the terminator, a sequence of DNA nucleotides that marks the end of the gene. Once it reaches the terminator, the RNA polymerase detaches from the DNA, restoring the original DNA double helix, and the newly synthesized RNA floats off.
So is the DNA changed at all by that process?
No.
No, which is awesome, right? Because if you need that same gene later today or tomorrow or a year from now, it's still there. So that's why transcription happens. It's a set of blueprints. A gene is a blueprint, and you may need to use that blueprint over and over and over. Okay? So that's an animation of kind of the general steps of transcription. What this more complicated figure is showing you here is that this is our double-stranded DNA molecule, right? So the RNA polymerase has pulled those strands open, which is easy to do because they're connected together by hydrogen bonds. So this is the DNA sequence. This is the new RNA molecule that's being made, okay? So what that means is when RNA polymerase comes up to a double-stranded piece of DNA and pulls it open, it's going to choose one strand to actually transcribe. It's not going to transcribe the other strand, right? That strand that's being used for transcription, so this is the DNA strand, right? This is the template strand. This is the one where RNA polymerase is actually looking at the sequence of DNA nucleotides and using those to assemble RNA nucleotides. So base pairing rules. So for RNA polymerase, we're going to make DNA black and RNA red. So if RNA polymerase is reading DNA, it may see an A, it may see a T, it may see a G, or it may see a C, right? When it sees that, it's going to put an RNA nucleotide, let's just make RNA general, following these rules. If RNA polymerase sees a D in the, or a D, sees an A in the DNA, it's going to put a U in the RNA, right? If it sees a T, it's going to put an A in the RNA. If it sees a G in the DNA, it's going to put a C in the RNA. And if it sees a C in the DNA, it's going to put a G, right? These are DNA nucleotides. These are RNA nucleotides, right?
So, and the RNA is the only one that has the U.
Yep, RNA is the only one that has a U. There is no U in DNA. There is no T, or sorry, there's no U in DNA. There, what did I do with? Oh, that's right.
So there is no U in DNA, like you said.
There's no U in
Oh,
DNA.
it's only in RNA.
Yeah, there's no T in RNA.
Yeah.
So how do you tell the strands apart from template and non-template? Is it the template just like the one where it's being, or is there a way to like determine which is which before?
That's a fantastic question. Here's the answer for what we're going to cover. I'm going to tell you which strand is the template strand.
Okay.
How does it actually happen in real life? It's the orientation of the promoter. RNA polymerase, when it binds to that sequence of DNA nucleotides, we're not going to get into the actual sequence of nucleotides, but we know what they are. They're pretty long and there's like different like blocks of them. But when RNA polymerase binds to a promoter, it orients it in such a way that it can tell which strand is the template strand. So the promoter contains that information. For us, I'm going to tell you which one it is. What's that?
Isn't the template strand the 5' 3'?
So if you think about this though, when you open this up, I mean, that's a good point, right? And we're going to go over the rules again for RNA polymerase, which are the same as DNA. So they have to follow the 3' to 5', right? But how do you know which strand to actually use? You would go in opposite directions,
Right.
but how does the enzyme know which direction to go in the first place? It has to pick one strand.
Okay.
And so what tells it that strand is the orientation of the promoter sequence.
Okay.
So we can get into promoters if you guys want to. Maybe not today. So this here is this template strand, right? Because this is the one that RNA polymerase, we're going to say this is the RNA polymerase enzyme. It's actively transcribing this DNA molecule by right now reading the template strand of the DNA. So if it sees a C in the DNA, the RNA is a G. If it sees a C, like that doesn't make any sense. They're not all lined up. These are. So if we see an A in the DNA, there's going to be a U in the RNA, right? This is the RNA molecule that's being built. That's our newly made RNA transcript. So if it sees a C in the DNA, it's going to put a G in the RNA. If it sees a T in the DNA, it's going to put an A in the RNA. So on and so forth.
That's what the active site is doing, or
Mhm.
the active site's the one making the new
Yep.
sequences.
The active site of the enzyme looks at this DNA and sees a T, so it finds an A RNA nucleotide and covalently links it to the last one.
So then the active site is like being carried by the RNA polymerase?
It's part of it, yeah.
Oh, okay.
So let me ask you this. So what's going to happen next in this picture is RNA polymerase is going to move to the next nucleotide. It's going to see the DNA is a C, so what's the next RNA nucleotide going to be? G. So that G nucleotide is going to get covalently attached right there. And then it'll go to the next one, right? So let's do this. So this is our double-stranded DNA molecule.
Did we say where the nucleotides
Right.
are coming from? Like, are they coming from the polymerase or are they coming from somewhere in the nucleus?
And I'm going to tell you that this is our template strand.
Right, so the new one where you're making ribosome, you see how like the U and the G are kind of like about to be added on? When we get outside the cell, they're gonna like come.
So when RNA polymerase is going to start transcription, first thing it has to do is pull the DNA strands apart. So I'm going to take a second and just do that. Right? Well, actually, let's start at the actual beginning. How's RNA polymerase even going to know where to start? Because the DNA is going to continue this way for millions of base pairs. It's going to go that way for millions of base pairs. So how are we going to know where to start?
The promoter.
The promoter. So this isn't really a promoter. I just put these 6 nucleotides over here. But we're going to say this is the promoter. And we're going to say that this right here with these is the terminator. So you see how these are nothing, there's nothing special about the promoter and the terminator from a structural point of view. There's no like proteins bound to it, nothing like that. It's just that theoretically, these aren't real promoter or terminator sequences, but the concept is there's just a sequence of nucleotides that RNA polymerase recognizes as a signal to stop, right? There's also a certain sequence of amino acids, a certain sequence of nucleotides that tells RNA polymerase this is where you start transcription for this gene, right? That's a promoter. So now if we're talking about E. coli, all those 4.2 million base pairs of DNA, RNA polymerase knows to come right here, right? And it's also, when it binds to that promoter, is going to be able to tell that this top strand is the one that we want to transcribe. It's the template strand, okay? Yeah.
So the promoter and the terminator are basically like, you said like sequences of nucleotides.
Yeah.
And it's like the same thing as a start and stop codon, right?
So no, because the start and stop codons are an RNA. So different. I mean, a different molecule. Same kind of concept though. It's this is where you start transcription. This is where you stop transcription. Now, when we have our RNA molecule, it's going to have a start codon telling the ribosome where to start, and a stop codon telling the ribosome where to stop. So same concept, just different molecule.
And it doesn't actually start until like the next one where it would be T.
Yeah, we're going to transcribe it.
Okay.
Yeah, for sure. So RNA polymerase has to know where to start, that's the promoter, has to know where to stop, that's the terminator, right? So RNA polymerase also has rules. But the nice thing is the rules in general are the same as DNA polymerase. So RNA polymerase is going to read the template strand (3' to 5'). And it's going to synthesize RNA 5' to 3'. Or I'll put make. Synthesize and make are the same thing. So once it figures out where the promoter is, that's where it's going to start. But what it has to do to start is separate these two strands of DNA, right? So I'm going to take a moment just to do that. 5'. Someone double check this. All I'm doing is recopying this lower strand. Is that right?
Wait, why the lower strand though?
So I just wanted to emphasize that RNA polymerase has to pull these strands apart. So it's the same exact strand, they're just now separated. So this strand is still completely intact because it's covalently linked together, all the nucleotides are. This strand is still covalently linked together because all the nucleotides are covalently linked together. But the hydrogen bonds have been broken, right? So the hydrogen bonds between the bases, like here, have been broken, right? So the first thing that has to happen for transcription is that RNA polymerase is going to bind to the promoter. We're going to say RNA polymerase is red, right? And it's going to see in its active site here that the very first DNA nucleotide is a T. So what's the first RNA nucleotide going to be?
A.
It's going to be an A, right? And then what's this one going to be?
C.
G. So if we stop there for a second, RNA polymerase binds to the promoter, so now it knows which strand to transcribe, which direction to go, because it's transcribing that template strand 3' to 5'. That's an important thing to remember, right? DNA doesn't matter if it's left or right, up or down. The only thing that matters is ends, right? So if this was a piece of double-stranded DNA, and I told you that this was the template strand, where would RNA polymerase bind?
To the 5'.
The one on the left, 3.
One on the left, right? Because RNA polymerase reads the template strand 3' to 5'. So if this was DNA and I told you that this strand was the template strand, always start at the 3' end of the template strand. So it would start right here. So don't get fooled by trying to read like a book, left or right, right? What if we did it like this? Let's see. So if I gave you a piece of double-stranded DNA that looked like that and told you the template strand was the top strand, where would RNA polymerase start? Here or here?
Right.
Start right here. So it has to follow that rule. It reads the template strand 3' to 5'. It doesn't matter left or right, up or down. What matters is number one, which strand is the template strand? Number two, where's the 3' end of it? Okay? So RNA polymerase went to this very first DNA nucleotide here, saw a T, so it put an A. Saw the second nucleotide, put a G, or it saw a G, so it put a C. It saw the third nucleotide was a C, so it put a G in the RNA. So our RNA molecule is now 3 nucleotides long. What end of the RNA molecule is this end?
3 prime?
So RNA polymerase reads template strand 3' to 5', and it makes new RNA 5' to 3'.
So that's the 5.
5' end. So the rules are very similar to DNA polymerase. So that is the 5' end of our RNA molecule. Now I drew RNA polymerase right here at the promoter, but remember it's moving down, 1 nucleotide at a time. I just don't want to draw that because it'd be a mess, right? But what it's going to do is just keep moving. It's going to keep reading that template strand 3' toward the 5' direction. While it's doing that, it's building a new RNA molecule. Remember, the RNA is red. It's building a new RNA molecule from its 5' end toward its 3' end. Yeah?
Where does it get the RNA nucleotides? Does it come from the polymerase or does it come from?
So in areas where, that's a really good question. And it also applies to ribosomes, where they get their amino acids, right? So inside, and they're very close to the areas of transcription, there are big pools of RNA nucleotides. So when you eat something or a bacteria ingests those, they come pre-made. But we and also bacteria can make nucleotides. So they either get them from the environment or they make their own or a combination of both. And at sites of transcriptions, there's just big pools of them by the millions, right? Yeah. And the same thing, right? When we get the translation, the same exact thing. There's 20 different amino acids that we have a choice to make proteins from. All a protein is a molecule, let's say 1,000 amino acids long. It's just the amino acids in the right order. Where does that giant pool of amino acids come from? And the answer for bacteria and us is either we bring them in or we make them. Some we can make, some we can't. So some we have to get in our diet. But the ribosome has to be surrounded by a massive pool of amino acids. RNA polymerase has to be surrounded by a massive pool of RNA nucleotides. What about DNA polymerase that we talked about last time? Same exact thing. It needs a massive pool of DNA nucleotides so it can grab an A when it needs it, grabs a T when it needs it, so on and so forth. Absolutely. So I'm going to keep transcribing this. So if this is a T, we're going to go U, and then we're going to see if it's A, we're going to put a U. If this is a C, we're going to put a G. If it's a G, we're going to put a C. So you notice anything funny?
You're
You
skipping
skip
the
the
T's?
I
And
didn't
there's
do it on
no
purpose.
start code on it? Or the
But do you
start
see
code on-
anything similar between this strand
Looks exactly
and
like
this
the,
strand?
Oh, yeah.
It looks the exact same except for the U and the T.
Yeah, so if we take this out, because this is part of our promoter sequence, if you look at the strand of DNA that's not the template strand, right, the other one, it has the exact same sequence as the template strand except for T's or U's, right? Which makes sense, right? Because this DNA strand is complementary to this DNA strand. And if we're going to make a RNA complementary to the template strand, it has to follow the same rules, except that there's no T's, there's U's. So that's why the other DNA strand is called the coding strand. So one strand is the template strand. That's the one that's actually being used by RNA polymerase. The coding strand is the one that makes it easier to figure out the sequence of an RNA if you're looking at the DNA sequence. Okay. So when you get this question, and I give you an RNA molecule, I tell you which strand is the template strand, and then you need to transcribe it. Remember that. You can just look at the other strand, the coding strand, and write down its exact sequence, except for you got to replace all the T's with U's. So someone tell me when I screw this up.
And the A's with the U's?
T's and U's.
Coded.
So take the T's in the DNA and make them U's. I'm having a hard time with this today.
You did it right.
No, it's just A, U, over here on the board.
U, P, C,
A.
and
You're
U.
good.
Yeah.
If finally figured it out. That all makes sense.
Why does it, you have it written up the opposite?
Is it written wrong over here?
A to U.
Yeah, A is
DNA.
to U.
So DNA has
Oh, I thought
C's and
these
G's.
lined up.
G binds to C. DNA has T's, right?
A.
So those are gonna be A's.
Yeah.
DNA has an A, which
Oh.
would bind if it was DNA to a T, but there's no, that's a good thing to keep straight. I just screwed it up, right? So that has to be a U, right?
Got ya. Okay, sorry.
No, you're totally good.
It just
I
has
just screwed it up
T's
too,
instead of U's.
right?
The non-template.
So when we're
It's
done
the
with
same as the
this,
non-template T.
RNA polymerase is transcribing along the template strand until it hits what? Stop codon. Terminator, and it stops. And just like the video we just watched, it just lets go, right? So if this is where RNA polymerase stops transcription, then what end of the RNA molecule is this?
That's the 3 prime.
3', right? So that is the RNA sequence that came from RNA polymerase transcribing that gene, right? So what you have to do, you have to get the double-stranded DNA molecule. You have to know which strand is the template strand. You have to go to the 3' end of the template strand, right? Find the end of the promoter. Start transcription. The very first RNA nucleotide is the one that's on the 5' end. Then you transcribe all the way down the length of the gene until you hit the terminator. Then you stop. The end of the RNA is the 3' end. Okay? So if we were going to keep going with this, which we are, the RNA, I'm going to draw this exact same molecule. Someone tell me if I screw it up. All right? And all I'm drawing it separate for is now the RNA molecule just completely lets go of the DNA, it lets go of RNA polymerase, and it floats away.
So we'll start at 5' again?
I'm just redrawing
Oh,
this.
okay.
Yeah. Just to kind of show you what happens next. So A, C, G, U, C, A, U, Q, U, talking to myself. G, C, A, U, A. So once we're done with this, right? This RNA molecule completely lets go of the DNA. So now it's left. And here it is. And the DNA, I'm not going to redraw this, but the DNA just simply binds back together. That's what I mean by these arrows. So at the end of this, the DNA molecule looks just like it did when we started. Both strands completely bound back together, closed up. We're done with it. If we want to make that, if we want to express that same gene, if we want to transcribe that gene later today, we totally can, or tomorrow, or next week, or whatever, right?
So after it transcribes, it goes back again.
Mm-hmm. Because the backbones are covalently bound together, super, super strong, all we did to separate it was break these really weak hydrogen bonds between the ATGs and Cs in the DNA. And so those were easy to break. So once our RNA molecule's done, it drifts away, and there's no reason those can't bind back together. So this thing was short. Let's watch this thing again.
Sorry, Mr. Casey.
It's not what I was really thinking to show.
So let's look at a single gene.
Let's play it.
I was like, oh, there's a soundtrack.
Let's look at a single gene, which consists of a stretch of DNA with some specific control sequences. Near the start of the gene is a region called the promoter. Near the end of the gene is a region called the terminator. The sequence between the promoter and the terminator is the part of the gene that codes for the protein being produced. Transcription begins with initiation, when a molecule of RNA polymerase binds to the promoter. The promoter contains a specific sequence of DNA bases that is recognized by the RNA polymerase, allowing it to bind. Notice that the sequence of the promoter plays an important role, even though it isn't part of the protein coding region of the gene. the RNA polymerase makes its way along the gene, synthesizing the RNA molecule from the template strand of the DNA. Elongation continues until RNA polymerase reaches the terminator, a sequence of DNA nucleotides that marks the end of the gene. Once it reaches the terminator, the RNA polymerase detaches from the DNA, restoring the original DNA double helix, and the newly synthesized RNA floats off.
See that? So here's our RNA. It's all by itself. Our DNA is bound back together. The hydrogen bonds are reformed. RNA polymerase is off doing something. Okay? So transcription is over now, right? But now what we've done is taken that language in DNA and we've transcribed it into RNA. So the next thing that we want to do is use this messenger RNA as a set of instructions on how to build a protein, right? So that is the next step. And so that's translation, right? So we're talking about translation a couple different ways. So first we're going to talk about not the mechanism of it, but in very, very general terms, what is going to happen. So, you guys remember what organelle is responsible for translation?
Ribosome?
Ribosome, exactly. So translation is the next step, right? And translation is the job of ribosomes. Okay? So ribosomes read a sequence of messenger RNA and use it as a set of instructions on how to build a protein. So if you think about it, the first thing a ribosome has to do is decide, okay, am I going to start reading the nucleotide sequence of the messenger RNA from the 5' end or the 3' end? And remember, that's the decision that the ribosome has to make. So on an exam, don't start thinking like you're reading like we normally do, left to right. That does not matter. So again, like we talked about before, if this is messenger RNA, which end is the ribosome going to start on?
Three.
It's going to start right here, right? If it looks like this. Because this is just a molecule. It could be all kinds of crazy shapes. Is the ribosome going to start here or here?
There.
Here, 5' end, right? Which is kind of counterintuitive because DNA polymerase and RNA polymerase started their jobs looking at the template strand and its 3' end and going towards 5' end. The ribosome starts at the 5' end. Okay? Always. You know, you could never say always, but like, if I was going to do it, I would say always. I mean, I've never, not even viruses do that differently. So, I think you can safely say always, right? So the ribosome is going to bind to the 5' end of this messenger RNA. Okay? So what the ribosome is going to do is move down the messenger RNA sequence from its 5' end toward its 3' end, and it's going to use the sequence of these RNA nucleotides as a source of information to tell it which amino acids to link together, in which order. So the next thing, the ribosome has to know where to start, right? So it's starting at the 5' end, but where does it start reading?
That second codon.
Yeah, exactly. But first, you guys know what codons are, right? Codons, I'm gonna tell you. So ribosomes, they bind to the 5' end of mRNA, and what they do is they look for a start codon. So the question is, first, what the heck is a codon? So I'm running out of room here. A codon is equal to 3 RNA nucleotides in a row. Okay? But it's not just random. So I would always give you this, or I will give you this on an exam. People spent a good part of the 1970s. Where'd you go? Here. So this is a chart I would always give you. right? So if you look here, what is all this stuff?
I freaking hate
Codon.
that stuff.
So if you look here, there's all these sets of 3. These are all codons, right? Right?
So living nightmare.
There's no fewer than this many, and there's no more than this many. So if you take the fact that at every single nucleotide position, you could have an A, U, C, or G, and codons are exactly 3 nucleotides long. If you do some statistics, these are all the different codons that are possible in a messenger RNA. There are no other ones. Okay? So in a codon is information that a ribosome uses to go from a codon sequence to an amino acid. And we know what and I always give you this, and we know what that code is. So if you look here, these 3 letters, these are all just 3-letter abbreviations for specific amino acids. 20 different amino acids, right? This is phenylalanine. The codon UUU and the codon UUC tell the ribosome to put in a phenylalanine. If you go here, if the ribosome sees a GUU or a GUC or a GUA or a GUG, that tells it, I need a valine. Okay? So if you keep going over here, right, well, let's go to here. If we see an AUU or an AUC, that's an isoleucine, right? So these are just all 20 amino acids. And what's weird is there's more than one codon that codes for some amino acids, right? But it has to be that way, because these are all the possible codons that can be in a messenger RNA. So thankfully, they all code for an amino acid, for the most part. If they didn't, the ribosome would get to a codon and be like, I don't know what goes there. And then that'd be bad, right? So all amino acids that are possible are on this chart. All codons that are possible are on this chart, okay? And I'll always give you this, right? So there's a handful of codons that act differently than most, right? So the first one is right here. You only need to memorize a couple of these codons, not the whole chart, obviously. So an AUG, anybody know what that is? Star codon, right? So AUG is what tells the ribosome to start translation. So the ribosome binds to the 5' end of the messenger RNA and starts looking for an AUG, because that is what the start codon is. So in other words, when the ribosome binds right here, it says, okay, that's an ACG. Nope, not a start codon. Moves down toward the 3' end, sees a CGA. Nope, not a start codon. Sees a GAU. Not a start codon, keeps moving down, sees an AUG right there. That tells it where to start translation. So if AUG is the code for the ribosome to put in a methionine amino acid, what is the first amino acid in every single protein? So methionine, because that's the only start codon, right?
It's a nut.
So this is what's called a 5' untranslated region. It's on the 5' end of the RNA, the messenger RNA, but it's not being translated.
What is
right?
it called?
5' untranslated region.
Untranslated.
Because it's not translated. It's not actually a protein sequence. It has a lot to do with the stability of the mRNA. We talked a little bit about that, right? So there's a lot of controlling of the life of the messenger RNA, even its localization, all kinds of cool stuff going on. So it's not like it's useless. It's not like it has no purpose. But as far as understanding the mechanism of translation, it doesn't do anything right now, right? So when the ribosome gets to that first AUG, that tells it where to start translation. It doesn't matter if there's no nucleotides here, if there's 3, if there's 1,000, if there's 942, it is completely irrelevant. The only thing the ribosome is looking for is that first AUG. And this is incredibly important, because this establishes what is called the reading frame, right? Because what's the next codon going to be?
You.
It's the next sequence of 3 RNA nucleotides. So this, let me fill this in a little bit. So this is the methionine, right? So the first amino acid in the primary sequence of our protein is going to be this methionine, right? All a protein is a sequence of amino acids, so what's number 2? Let's figure it out. Ribosome goes to the next codon, sees a CAU, and so what does it decide to do?
Histamine.
Puts in a histidine, right?
Histidine.
So if you look here, you can either I never use these keys at the top and sides, but you can. But this here's the first letter. So if we're looking for a CAU, these sides are meant to be used like this. So okay, so C, so what we're looking for is in this row. A, second letter, so CA, it's somewhere in here. And then U, and you can narrow it down that way if you want to. Or you can just look at that, it's not that big, right? Whatever you want to do. So regardless, that's our second amino acid. So now our protein's primary sequence is a methionine covalently linked to a histidine, right? So what's our next codon? What does that tell the ribosome to put as amino acid number 3? What is it? Is that a phenylalanine? UUU? Phenylalanine, right there. Yeah.
Can we get
That's all
a
it says.
bank word for the abbreviations?
You're gonna always have this chart.
No, but that doesn't say
But,
like
to
what
say-
they are. We're just guessing because we only
Yeah,
got the
actually,
first
yeah,
two.
absolutely. So do any of these look familiar? Like, do you guys like proline, arginine, tryptophan? You guys ever learn those words?
GLU and GL-Y.
This is glutamine and glutamic acid.
Glutamine, glutamic acid.
What's the glide?
Glycine.
Okay.
Yeah. So
Aspirin?
so now our protein is 3
Aspirin.
amino acids
Yeah, right
long.
there.
What's our next codon?
GCA.
Glucose.
What amino acid?
Alan.
That's
Methylphenidate.
ALA.
Alanine.
Glucocytosis.
So now our protein is methionine, histidine, phenylalanine, alanine, right? So now the ribosome is going to go to the next codon. What does that code for?
That's a stop codon.
That's a stop codon, right? So there's 3 different stop codons. Why are there 3? I have no idea. Why are there 4 on here? UAA, UAG, and UGA. I don't know what that's about. I won't give you that chart. So UAA, UAG, and UGA are stop codons. That's why I put this one right here.
Is that why there's confusion? Like if you Google how many stop codons are there, they'll say sometimes they'll say there's 4, sometimes they'll say there's 3.
Really?
Which one's correct?
I did not. Well, it's 3,
Is it?
unless there's a new, which could be.
I
There's always strange organisms being discovered that do things differently. So I'm gonna Google that because I'm really curious now. Yeah.
Y'all believe what the Google says?
So if I read it online, it has to be true. So when a ribosome hits a stop codon, what amino acid is it code for?
nothing.
Nothing. It doesn't. It doesn't. And this is one thing that always happens when I give this question on an exam. Remember that a start codon codes for an amino acid, which is always a methionine, and a stop codon never does. Okay? A stop codon is a signal to the ribosome hey, you're done. So what's going to happen is it's going to fall apart. I keep drawing the ribosome as 2 pieces because they're 2 giant subunits. But when translation's over, the 2 subunits fall apart, the messenger RNA goes away, and our protein starts to fold up into its secondary structure, and then its tertiary structure, if you remember that from your prereq, and then it's ready to go, right? You see why DNA mutations may, not always, but may cause proteins to be made incorrectly. Because if there's a mistake in the DNA sequence, then I might have a mutation in the messenger RNA sequence that would result in the incorrect amino acid being put into a protein. But do you also see why sometimes DNA mutations happen that don't hurt the protein at all? Because what if I had a mutation that made a U into a C right here? So if I changed a GUU codon to a GUC, would that mutate the protein?
No.
No. The DNA mutation would result in a mutation in the messenger RNA, but if 2 codons code for the same amino acid, I'm getting the exact same protein. We'll talk more about mutations next week. Okay?
I had a dozen questions about
It can.
mutations.
Yeah. Sickle cell anemia is caused by one mistake in a DNA nucleotide that makes amino acid number 7 of hemoglobin wrong. That's all it is. But that can kill you. Someone have a question?
No, I said, what, cause that's what sickle cell is, it's one amino acid that's wrong in the DNA.
It's not even missing, it's just one of them is incorrect.
Oh my God.
And that's just
And people
purely
go
by
through that much pain.
chance?
My patients is.
Yeah, it's a long, complicated story. There's an evolutionary pressure that keeps it in. Let's go down that road for a second. So people who
Spit it out, you got it.
you guys know what heterozygous is?
Heterozygous.
So if you have one good copy of a gene and one bad copy, so if you have two bad copies of a hemoglobin gene, you're in big trouble, right? If you have two good copies, you're never going to have sickle cell anemia. If you're heterozygous, meaning you have one good copy and one bad copy, you can suffer from sickle cell anemia, right? But here's the crazy thing. If you have a bad copy of that hemoglobin gene, you are resistant to malaria. So any place in the world where there's a high prevalence of malaria, you find a high occurrence of people having the gene, the mutated sickle cell gene, because it makes them more resistant to malaria. So think about this. If you were living in a place that had a high amount of malaria, right, and you and your partner were heterozygous, meaning you both have one good copy of the gene and one bad copy. If you had 4 kids, 25% of those kids would be homozygous dominant for that mutation, which means they'd be resistant to malaria, but they'd get sickle cell and probably die pretty quickly, right? 25% of their kids would have 2 good copies of the gene. So they'd never get sickle cell anemia, but they're super susceptible to malaria. 50% of them would be heterozygous. So they would have sickle cell a little bit, but they'd probably be pretty much okay, and they'd also be resistant to malaria. So 50% of, sounds terrible, but from a biology point of view, if 50% of your offspring make it, that's really good, right?
That just goes back to like Punnett squares and stuff, right?
Mm-hmm. Exactly, Punnett squares. Yeah. So, but because that mutation, which can hurt you, especially if you're homozygous dominant, also gives you an advantage in some particular situations, that allele, that mutated allele, has stayed with us for a long, long time. And if you look at people as populations, if you come from a place with a high prevalence of malaria, you are much more likely to have that gene because it protected the people that you came from. So, anyway. Okay. So, any questions on this? So what I want to do now is we're going to watch an animation, because now we got to talk about how this happens mechanistically, right? What I mean by that is how the heck does a ribosome have the ability to see a sequence and messenger RNA and actually know which amino acid that it needs to add? So how is that going to happen? So I'm going to watch how is it so late so quick? We're going to watch an animation, and then we're going to talk about that animation in detail starting next week, right? But I want to plant this kind of information in your heads. So this is not going to look super familiar, but just go with it, right? So this is the ribosome, large subunit here, small one. This is our messenger RNA, and it won't be this blurry when it starts playing. These are all amino acids being linked together in a row to build a protein just like this. These structures are here, something we haven't talked about. These are transfer RNAs, right? These are RNA molecules, and their job is to link information in messenger RNA with amino acids, right? This was mind-boggling to me. Like, my first major was pre-law. I'm so glad I'm not a lawyer, right? Cut my hair, wear a suit. When I first learned this, I was like, the first time ever in college, I was like blown away by something. Like, it's just cool. Like, we know how to look at DNA, and we can figure out anything's DNA now. And we can tell exactly how it's put together. And then we can change it, which all kinds of implications coming along with that. But it's just amazing to me.
So you said the transfer RNA is messenger RNA to DNA.
Let's just watch this video.
Okay.
Then we'll talk about it next time.
What do you say? Are we coming done? Oh. Looks like SpongeBob. Doesn't it?
Prokaryotic cell
So, actually, we'll just watch it and then we'll do it.
Yeah, it's just.
Translation is initiated by formation of an initiation complex consisting of the 30S ribosomal subunit, formal methionine tRNA, and messenger RNA. The 50S ribosomal subunit then joins the complex. Proteins called initiation factors are also involved, but are not shown. The 70S ribosome has two sites to which transfer RNA carrying amino acids can bind. One is called the peptidyl or P site, and the other is called the acceptor or A site.
Bless you.
There is also a third site called the exit or E site, where transfer RNAs are released. The initiating transfer RNA carrying formal methionine binds to the P site. A transfer RNA that recognizes the next codon and carries the second amino acid then moves into the A site. The formal methionine carried by the transfer RNA in the P site is then joined to the amino acid carried by the transfer RNA that just entered the A site by a peptide bond. The ribosome now advances a distance of one codon, and the transfer RNA that carried the formal methionine is released at the E site. A transfer RNA carrying the next amino acid now moves into the A site, where the anticodon on the transfer RNA matches the codon on the messenger RNA. The ribosome shifts down by a distance of one codon. As the shift occurs, the two amino acids on the transfer RNA in the P site are transferred to the new amino acid, and the second transfer RNA is released from the E site. The ribosome continues to move along the messenger RNA, and new amino acids are added to the growing polypeptide chain. Elongation of the polypeptide is terminated when a stop codon moves into the A site. A stop codon does not specify an amino acid and does not have a corresponding transfer RNA. The ribosome dissociates into the 30S and 50S subunits, and the messenger RNA and protein are released.
Okay, so I realize a lot of that was new, but we will talk more about transfer RNAs and how they're working and fill in the gaps on translation on Tuesday. So have a good weekend, you guys.
Can you believe that